Alright, so today we're going to look at muscle contraction. Muscle contraction can be broken into three phases just to try and simplify the process. In actual fact, muscle contraction is just one complete process. Once the muscle is stimulated, it's an all or nothing response. But in terms of us understanding the actual pathway, if you like, of the physiological actions, then having the three phases is much more beneficial in terms of our understanding it and learning. So in this first one, we're going to look at the first phase, excitation. On the board behind me, I've written the three phases, and the first one's got a tick to it, so that's excitation. That's what we're going to look at. And we're going to try and keep it step by step and keep it simple. And remember, in muscle contraction, we're going to look at all of the combined pieces of information that we've been looking at for the first 4 to 5 weeks in physiology, including the importance of proteins and their ability to change their shape. The reasons why they change the shape are due to charges, and charges can be introduced to a protein either by chemical bonding of another molecule attaching to the protein, bringing charge with it, or it can be just with a charge associating around the protein structure itself, such as a charge buildup. Okay, so let's get started. So behind me here, I have the diagram I've already drawn out. This part here is the axon terminal of the neuron, and the action potential arrives in the axon terminal and is converted into a chemical signal. The chemical signal, in this case in a neuromuscular junction, is a neurotransmitter called acetylcholine, shortened to ACH. So acetylcholine is released across the synapse, which is between the axon terminal of the neuron and the surface of the muscle cell. So the action potential arrives, a little bit of physiology occurs in here, including calcium channels. ACH is released, and it moves onto the surface of the muscle, called the sarcolemma, by the action of diffusion. And it finds binding sites on special protein carriers, and these protein carriers are sodium, potassium, chemically gated protein channels. Okay, so there are two binding sites on each one of these protein channels. So once an ACH is bound here and an ACH is bound here, the channel opens up, and that allows sodium to flow in, bringing with it positive charge. But this is a sodium-potassium channel. So you also have the movement of potassium flowing out. Now, keep in mind a couple of things about the cell. First of all, the cell at rest has a positive charge around the outside of the cell and a negative charge inside of the cell. The positive charge outside of the cell is created by a very large concentration gradient for sodium. Now that concentration gradient is maintained by the presence of sodium-potassium pumps on the cell membrane, and that pumps out 3 sodium ions and pumps in 2 potassium ions. So sodium builds up on the outside and potassium builds up on the inside. So why is the inside of the cell negative when potassium is building up inside and it's positive? Because all the chemistry taking place inside the cell is producing predominantly negatively charged species. And so those negative charges build up and create a polarization across the membrane. Positive outside, negative inside. So in order to create a change in the surface of the muscle, in this case the muscle, on the surface of the cell, you have to depolarize the membrane. So remember, positive outside, negative inside. So in order to create the change, open up a channel, allow positive charge in, and that will create positive on the inside as well, therefore taking away polarization, which in other terms is depolarization. So, remind yourself, this is normally negative, and on the outside positive. So when this channel opens, sodium flows in, but potassium flows out. Now the sodium gradient is about 10 times stronger on the outside than it is on the inside. Therefore, when the sodium channels open up, sodium is going to flow into the cell and quickly. The potassium concentration gradient is weaker than the sodium gradient, only about 4 times stronger on the inside than potassium on the outside. So although potassium flows out taking positive charge with it, it doesn't flow out and take as much positive charge as that is entering because of the sodium coming in. So overall we end up with a positive charge build-up on the inside of the membrane. So we've had our action potential from the neuron, our neurotransmitter, acetylcholine, has been released, it is diffused across, it has found binding sites on these chemically gated protein channels. And these channels are called nicotinic cholinergic receptors. Sorry, that writing isn't very clear. Nicotinic cholinergic receptors. Once 2 of these acetylcholine molecules have bound to the protein channel, it opens, sodium comes in at a greater rate than potassium leaves, and it creates a positive environment around where the channel has opened. That positive environment builds up on the inside of the membrane, and this is its target. So we've had our stimulation from the neuron, acetylcholine's been released and bound to the muscle cell membrane, the chemically gated protein channel has opened, sodium ions have entered. This has caused a depolarization of the muscle cell membrane, or sarcolemma to use its proper name, and this depolarization event causes positive charge to build up on the inside of the sarcolemma. As it builds up, it diffuses out because positive charge rejects itself, and it gets closer and closer to this channel here. Now this channel is a sodium voltage gated channel, so the build-up of positive charge on the inside causes it to change its shape, and it's a sodium channel, so sodium flows in, bringing with it more positive charge, more depolarization, opening up the next sodium gated channel. That voltage gated channel opens, causing a domino effect of positive charge, opening up sodium channels all the way down. And that creates all that positive charge on the inside, and whilst it's doing that, that positive charge is causing the opening of calcium voltage gated channels on the sarcoplasmic reticulum. The sarcoplasmic reticulum is a modification of a normal smooth endoplasmic reticulum, and its job is to store high concentrations of calcium. It does that because there are calcium pumps built into the membrane, and they pump calcium in to the membrane at rest. So this positive charge, starting from an action potential in the neuron, being converted to a neurotransmitter acetylcholine, opening up a chemically gated channel here on the surface of the membrane, a nicotinic cholinergic receptor, 2 acetylcholines bind, it opens up, sodium comes in, potassium goes out, positive charge builds up on the inside, positive on the outside, that's a depolarization, there's no polarization there. That causes the first voltage gated channel to open, it's sodium, and that causes a domino effect of sodium voltage gated channels opening down the surface of the sarcolemma and down into the T-tubule, deep into the cell itself, and that results in the opening of calcium channels. And calcium channels release calcium all over the myofibrils. And that is the end of excitation. Thank you for listening.